Orientation to biotechnology. This PowerPoint video will cover Chapter 1, Intro to Biotech. We ask, what is biotechnology? How does it impact myself and others? And why do most people know so little about biotechnology? Biotechnology defined. It's the science of using living organisms or their products for the benefits of humans. Most people use products produced through biotechnology without realizing it, including you. Some of these products are medicines, food, and energy to save or improve our lives. Here we see a medicine, Zofran or ondansetron hydrochloride injection. This looks like a 20 ml bottle. Could be either single use or multi-dose, and this would be injected into a patient at a hospital or an outpatient clinic for, acute nausea. So that should help settle the stomach, just from my knowledge of being a pharmacy tech. We have corn. I'm not sure if this is corn here or something, but looks like it could be. I need to study agriculture and see if this is like wheat or corn. I don't see any corn stalks though. But then we have biodiesel powered by Propel fuels. So this is at like a gas station where a truck could fill up, or diesel fuel could be given. So we get all these from biotechnology. We just don't know it. Biotechnology is actually thousands of years old, making cheese, wine, beer, bread, and the selective breeding of plants and animals are all examples of using living organisms for our benefit. Fermentation. Many common food products are a direct result of fermentation by microorganisms. We have yeast. Yeast ferment sugars to produce bread, rises from carbon dioxide gas or CO2 gas. So yeast ferment sugars to produce bread and beer and wine. Ethanol byproducts, which are the beer and wine. Lactobacilli ferment dairy products into yogurt and cheese through lactic acid fermentation. Any other foods that you can think of that are produced in this way? I can't think of any. Maybe during class I can come up with some ideas. Selective breeding. Selective breeding selects or chooses the organism with the most desirable traits to breed for the next generation. Another term for this is artificial selection. We've seen this in some biology classes, some intro to biology. And that was recently mentioned in organismal biology. This is often seen in animals and plants. Can you see what corn originally looked like on the left side compared to modern corn on the right? Wow. So that's really different, huh? So this is what corn originally looked like, and then compared to the modern corn on the right, this is what we see. What traits were selected for? What holds the information for the traits? Well, it looks like if these are the individual kernels, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Yeah, it just seems like as they got larger and larger, they were able to produce more and more kernels. Selective breeding continued. We can also see the selective breeding of zebrafish from normal to transparent, Casper variety. So this is B and this is C. Is there any human benefit to having a transparent zebrafish? From normal to transparent Casper variety. Maybe we can answer this question throughout reading the chapter. Is there any human benefit to having a transparent zebrafish? Antibiotics and vaccines are other products of biotechnology that has helped eradicate or control many types of infections and diseases, allowing humans and animals live longer, healthier lives. Where do antibiotics and vaccines come from? Antibiotics. In 1928, Alexander Fleming notices mold contamination, Penicillium notatum, on his bacterial petri dish of Staphylococcus aureus. He realizes that the mold must be producing a substance that inhibits the growth of the bacteria. This insight ushered in the modern era of antibiotics, a penicillin-type antibiotic disc inhibiting bacteria. So we see here the penicillin-type antibiotic disc, and the bacteria are here. So as we see in this middle section, there aren't as many, there's nearly no bacteria around, and as we get further and further from this penicillin disc, then we see the bacteria. The bacteria is here around the outer edges, right? So as it gets closer to this the penicillin disc here in the middle, it's resistant. The growth is inhibited, right, because of this penicillin disc, and that's the antibiotic. Penicillin was one of the first large-scale pharmaceutical products produced from a living organism in the 1940s. Being able to supply sufficient amounts of antibiotic in World War II greatly reduced the death toll from wounds and may have been a deciding factor in the war. Eli Lilly, Abbott, Merck, Pfizer, and Squibb were the main producers. At the top, we have an advertisement for penicillin. At the bottom, Fleming inspects a large penicillin fermenter. Interesting. So working in the pharmacy, I have experience with each of these pharmaceutical manufacturers. Modern biotechnology. What do you do if there are no organisms that produce your drug naturally? Can we force an organism to make a product it normally doesn't make? This is where genetic engineering and recombinant DNA technology come in. We've known since the 1950s that DNA is the source of genetic information, and Watson and Crick revealed the structure of DNA. Most biotech products are protein in nature, and it is DNA that codes for proteins, as we learned in cellular biology. So if we alter an organism's DNA, a gene to be specific, it hopefully will produce our protein of interest. So these go back to those questions here. Can we force an organism to make a product it normally doesn't make? And what do you do if there are no organisms that produce your drug naturally? So we can alter an organism's DNA, and hopefully it'll produce the protein. Example is people with type 1 diabetes produce little or no insulin, and they require insulin injections to maintain the proper glucose level in their blood. Patients use horse and pig insulin as an insulin substitute, but it was never quite as good as human insulin. The idea, what if we take a gene for human insulin and insert into a bacteria and have it produce the human insulin protein for us in large quantities? This is a common technique used to produce many modern biotech drugs, and we're going to see in figure 1.4. Figure 1.4 in A. The gene of interest lies here. To produce or introduce into bacterial or mammalian cells, the cells grow on culture plates. Grow cells that have gene of interest in fermenter. Cells cultured in fermenter produce the protein of interest. So here we have 1, 2, 3, 4, 5 cells with the gene introduced into bacterial or mammalian cells. So these two cells are the either the bacterial or mammalian cells. The cells grow that have a gene of interest, right? They reproduce, and now we see all of them have the gene of interest. Recombinant protein of interest can be isolated from cultured cells using biochemical techniques. So they were able to remove this recombinant protein of interest from these cells by using biochemical techniques. Okay. And here's an example of a finished product, Humulin R. And we've dispensed this out in the pharmacy. Modern biotechnology. Human insulin is manufactured in this way using bacteria and was one of the early big biotech drugs produced by Genentech in the 1980s. And this picture here is a recombinant bioreactor used for making synthetic human insulin. There are many types of or disciplines within the biotech industry. They all require a strong foundation in science, no matter which branch you pursue. We will touch on many, but not all of these, as biotech has become ever more diverse. See Figure 1.3. Figure 1.3, we have applications. Biopharmaceutics, pharmaceuticals and healthcare, biopharma, gene therapy, gene knockout testing, fermentation technology. This is towards the top. We have regulatory approval oversight, Food and Drug Administration, FDA, Environmental Protection Agency, EPA, U.S. Department of Agriculture, USDA, OSHA, Occupational Safety and Health Administration, drug development, bioinformatics, high throughput screening, cell culture and human testing, environmental and aquaculture, bioremediation, transgenic modifications, bioextractions, agricultural biotechnology, transgenic crops, DNA tracking of seeds, transgenic animals, detection, forensics, DNA fingerprinting, disease DNA detection, species member ID, genetic engineering through molecular biology, and analysis through bioinformatics. We have computer science, chemical engineering, physics, genetics, biochemistry, microbiology, statistics, immunology, molecular and cell biology, mathematics, human, animal, and plant physiology. Biotech companies. There's often confusion between pharmaceutical companies and biotech companies. Big Pharma has been around a long time, but they typically do not use living organisms. Some are starting to, though. They do not use living organisms, but they produce drugs chemically or synthetically. Biotech, on the other hand, uses modified organisms to produce a biologic product and are typically smaller than Big Pharma, although the gap is closing. Table 1.3, top 5 biotechnology companies and top 5 pharmaceutical companies by revenue. Biotech companies. So the revenue in millions. Amgen, 14.26 million. Genentech, now part of Roche, 11.72 million. Genzyme, UCB, Gilead, 3 million. Pharma companies. Now you can see exponentially how many millions that these have. Pharma companies: Johnson & Johnson, 61.89 million. Pfizer, 50.0 million. Roche, GlaxoSmithKline, Novartis, 44.26 million. So these big pharma companies have been around for a lot longer, and Amgen is a company that resides. They have a plant in Rhode Island, actually. Some biotechs are in our backyard. Amgen, like I mentioned, West Greenwich, RI, Alexion, Smithfield, Rhode Island, Neurotech, Cumberland, Rhode Island, Nebsis, Providence, and Bard, Warwick. And I'm sorry, so this actually, there was some notes that we needed to touch on. So I'm sorry to go back, but using genetically modified cultured cells to make proteins of interest. So genes of interest can be introduced into bacterial or mammalian cells, and such cells can be grown using cell culture techniques. Recombinant proteins isolated from these cells are used in hundreds of different biotechnology applications, but in this particular example, these are mammalian cells, okay? But the process is also commonly carried out using bacteria. And for this slide here, the notes, the biotechnology tree. Disciplines or different disciplines contribute to biotechnology, and we were discussing just the labels on here, the different disciplines. The basic sciences are the foundation or the roots of all aspects of biotechnology. So these are basically like the undergraduate classes or the required classes that you would have to study in order to get into the main biotechnology. And they were human physiology, animal plant physiology, mathematics, molecular and cellular biology, statistics, microbiology, genetics, biochemistry, physics. All these courses here, you're going to at least have to take a course in one of these to get involved in biotechnology. The central focus or the trunk for most biotechnological applications is genetic engineering. Branches of the tree represent different organisms, technologies, and applications that stem from genetic engineering and bioinformatics, central aspects of most biotechnological approaches. The regulation of biotechnology occurs through governmental agencies like the FDA, EPA, USDA, and OSHA. So those are more studied in Chapter 12. And here, investigational biotechnology drugs by disease category, Table 1.1, Figure 1.5. Table 1.1 is next. But here we have the production of drugs to combat cancer dominates the biotechnology industry's interest in treating human disease, with AIDS-related research and treatment of infectious diseases such as flu also near the top of the list. So, as we can see, cancer, AIDS, infectious diseases, these are where a lot of the work is being focused on, the production of the drugs. So if I plan on getting a Bachelor of Science in Biotechnology Manufacturing, I'm most likely going to work for a life science company that is focusing on one of these top 5, right? But it could be in one of these here. So it's good to see initiative in all of these different diseases. Table 1.1, we have top 10 biotechnology drugs with sales over $1 billion. Table 1.1, we have drug Enbrel. So we know this developer at Amgen and Wyeth function rheumatoid arthritis. So this drug we sold a lot of in the pharmacy, or we didn't sell it, but we dispensed a lot in the military pharmacy. We don't handle money there, but we had a lot of patients that were on this drug, and I handled the pharmaceutical supply for the department in 2 pharmacies. And this drug Enbrel was so expensive. It was one of the most expensive drugs that we kept in the pharmacy, and it would have to be refrigerated. There would be maybe a box of maybe 14 applications or administrations. It would be like a month's supply would be about $1,500 to $2,000. So a year's supply of this would be, it would easily cost $12,000 for somebody to have a year's supply of this. And people were definitely getting it. And they were very upset when this Enbrel was not available. Sometimes it would be out of stocks. Sometimes it would be on back order. It was really a in-demand item. Very, very expensive item. Remicade by Johnson & Johnson for the same rheumatoid arthritis. I wonder how the treatment efficacy compares to these 2 products. Rituxan by Roche, non-Hodgkin's lymphoma. Avastin by Roche, colon cancer. Herceptin for breast cancer. Humira for rheumatoid arthritis. That's another very expensive and very common dispensed medicine within the pharmacy. Levinox, Sanofi Aventis for blood clots. Lantus. This was extreme. We dispensed so much of this Lantus insulin pens. They were pre-filled syringes, and would have like a 5-pack, and the user would be able to manipulate the pen in such a way where they were able to dispense the correct amount of units for their treatment, for their therapy. Aranesp by Amgen for anemia. So all these products, they're not cheap. None of them are cheap. And I would say for the most part, at least I know Enbrel, Humira, Lantus, these were refrigerated. You had to have them refrigerated. I haven't had much experience with the others, but I bet that those are probably in the same kind of boat, because they're biologics, so they can't really be exposed to high temperatures. They have to have safe, strict handling. Table 1.2, we have examples of proteins manufactured from cloned genes. And product, so we have blood factor 8, clotting factor, treat hemophilia, epidermal growth factor, used with application is to stimulate antibody production in patients with immune system disorders. Immune system disorders, their antibodies are being suppressed, so they need this growth factor to help produce more of them. Growth hormone, correct pituitary deficiencies in short stature in humans. Other forms are used in cows to increase milk production. We have insulin to treat diabetes. Interferons treat cancer and viral infections. Interleukins treat cancer and stimulate antibody production. Monoclonal antibodies diagnose and treat a variety of diseases, including arthritis, and cancer. Tissue plasminogen activator. Wow, my father just had this 2 days ago. He had a stroke 2 days ago, on Saturday night, the day before Trump met Kim Jong-un at the DMZ in North Korea. He had to get tissue plasminogen activator, and when he had his stroke, luckily he was with his girlfriend, and they were able to administer it within the window. They have a 4-hour window from the onset of the stroke to be able to administer this tissue plasminogen activator, TPA, and to treat heart attacks and stroke. And it's a godsend that they have this product, because it helped avoid the further building of the blood clot that was in his brain. That's what the TPA does. It prevents the blood clot from being able to get larger. So they mentioned that it was a blood thinner. Here we have figure 1.16, distribution of United States biotechnology companies. Public and private biotech companies are located throughout the U.S. We have California has the most, over 350. Wow, Rhode Island's not even on the map. Massachusetts has 250. So, fortunately, that's on both sides of the U.S. We have West Coast with Cali, and then Massachusetts is on the East Coast. And then these here are somewhat in between. We got North Carolina, Maryland is on our end, New Jersey, Pennsylvania, Georgia, New York, Texas, Florida, Washington, Connecticut. So those are, we pretty much, depending on the spread in California, it looks like the East Coast dominates pretty much. Yeah, because let's go back. So we have about 50, so 50 per. Yeah, it looks like 250. We have Maryland, there's a little, probably like 75. Jersey's over 100. So yeah, if we added up all those East Coast locations, I'm sure it definitely, en masse, it definitely exceeds what California's got. So this looks like there's more opportunity for biotechnology on the East Coast than there is on the West Coast, but I could be wrong. It's just a rough assessment. Here we have organizational structure of a typical medium-sized biotechnology company. Figure 1.17. Biotech companies range in size, but most companies have organizational structures similar to those depicted in this figure. Notice the range of different aspects of a company, from research and development, R&D, through sales, marketing, and legal aspects of a product. I don't know why it's automatically advancing to the next slide. Research and development, preclinical discovery research, bioinformatics and lab safety, operations, process, product development, manufacturing and production, quality control and assurance, clinical research and regulatory affairs, and we have finance and administration, business administration, development, information systems, legal and facilities management. So we have all these different jobs here that these here, the product and process development, manufacturing, production, these are some of the jobs that I would be interested in. So it could be calibration tech, manufacturing instrumentation, manufacturing technician, operator, manufacturing associate, manufacturing supervisor. And that's the manufacturing production side. Process development and product development. Director of process and product development, supervisor, associate, and then the technician. Logistics, and that's where we're going to be doing the shipping, customer service, distribution, operations, and transportation. Then you can see on research and development side, we have, these are the lab facilities. We have manager, supervisor, animal sciences, veterinarians, lab assistants, glass washers. Bioinformatics, we have the scientists, engineers, analysts, programmers, molecular modeler. And then moving up the chain, research associate, senior research associate, scientist 1 and 2, senior scientist, principal scientist, associate science director, or scientific director. These are the more higher paying jobs. So as you look up here, VP of Research Development, these are the highest paying jobs, obviously. Quality control, you have quality assurance, QA manager, supervisor, documentation specialist, documentation coordinate associate. Moving up there higher, microbiology, QC analyst, quality control analyst, chemistry, QC technician, analyst, quality control. Regulatory affairs, you have the clinical data manager and associate, regulatory affairs associate, and the manager of that department, clinical research manager. And then on the finance and administration side, these are where sales, marketing, medical information, shipper, receiver, facilities technician, facilities manager under the facilities management. Then on the legal side, we have patent and intellectual property, or IP attorney, very important process and part of the production part. Information systems manager, the information systems analyst, programmer. Director of human resources, human resource representatives, safety managers, purchasing agent and buyer, receptionist and administrative assistants. Director of business development, and then we have chief financial officers, accounting managers, accounting clerks, and internal auditors. And of course the VPs of finance. Next we have careers in biotechnology, manufacturing technician. So we see one here using some instrumentation, and this looks like it could be a bioreactor. It's got a really it's got a lot connected to it. Lots of gauges, lines, different types of lines, that have to be cleaned, right? Manufacturing techs, the critical employees, they work directly with the product, requires troubleshooting and some mechanical abilities. Quality control technicians, QC techs, they work with samples and lab equipment to ensure the product integrity, requires concentration and problem solving. Research assistants design experiments and makes detailed observations, analyzes data and interprets results. And here we have figure 1.8, minimal level of education required of entry-level technicians. The resource group performed a survey of entry-level education requirements for 69 biotechnology companies in the 3-county area of Central Coast, California. Results are comparable with other areas of the United States and indicate that R&D, research and development, generally requires a greater amount of training than other job areas. Of course. So we have analysis, production, QA, QC, and R&D. So we have the lightest, or it looks like the lightest blue, the R&D. So 6.7% high school, and high school. We have associate in arts or an associate in science degree, or certificates, BA or BS degrees and certificates, and graduate degrees. So research and development requires graduate degrees, or most jobs with R&D require a graduate degree. There are not too many production jobs that require a graduate degree. As compared to the bachelor's of science. So QA and QC looks like the bachelor's degree is a really good option. Bachelor's degree, you can't go wrong, and that's what I'm trying to pursue, is a BS, Bachelor of Science in Medical Lab Science and Biotechnology Manufacturing. So I'll be able to try and target some of these jobs here in analysis, production, QA, QC, and research and development. 60%. So that's a percent of businesses, 60% will hire you. 60% of businesses will hire you for a research and development position with a bachelor's degree. 46.9% of businesses in QA and QC, quality assurance or quality control, will hire you with a bachelor's degree. So you can see these percentages are lower, significantly lower here. Production. So production, they'll start you off. I'm surprised, but I guess I'll learn more as we go. Yeah. Bachelor's is the way to go. Because these jobs in analysis, QA, QC, and research and development, these are the jobs that I do want to get. I don't really want to focus in production, but you got to start somewhere. So, and that concludes the PowerPoint slideshow for Chapter 1 of Biotechnology.